All right, so we are going to move into transcription and translation. So our textbook does a whole chapter on each one. Whereas like the biology 101, we just go one chapter for both of them. So we're going to go through the whole process of gene expression. So first we're going to define a gene, and then we're going to talk about how we get the information from the gene to the phenotype of the organism. So generally, this is going to mean that we're going to produce a protein, but we're also going to talk about some other options besides just making a protein out of the information in our gene. So before we get to that, we need to go over the structure of RNA. So at one of the handouts I gave you is a comparison of RNA and DNA. So generally the nucleotides are the same. There's one nucleotide different between the two, and then we'll talk a little bit about some of the other differences. But one of the things that our textbook goes into at this point is a little bit deeper discussion on RNA and the importance of RNA, and why RNA is used in our cell. So if we look back to the first living organisms, which we didn't cover origin of life in this class, but as you start learning about what the first living organisms needed to look like, they had to have a few very specific characteristics. And one of those was a coding molecule, a molecule that could hold genetic information and then express that genetic information. Part of that is that the molecule also had to be replicatable. So there had to be a way for early organisms who didn't have a lot of stuff going on in their early cells to be able to actually replicate and pass on their genetic information. So early cells had to have a membrane, early cells had to have some organic molecules, and early cells had to have a coding molecule. So the leading hypothesis is that that coding molecule was RNA. So today our living organisms use DNA as their main information storage molecule. But there's a couple of reasons why RNA was probably used first. And if we think back to DNA replication, we needed two things, basically. We needed DNA and we needed proteins in order to replicate that DNA. So those proteins come from the DNA. The proteins for replication are coded from genes in the DNA. So what we have is kind of a circular argument here. Did DNA come first or did proteins come first? Because if proteins came first, then the coding molecule had to evolve after. But if DNA came first, then how did we replicate it if there were no proteins? So the hypothesis then is actually that RNA was the original coding molecule. And the reason for that is that some RNA molecules can act as ribozymes. So a ribozyme is an RNA molecule that can act as an enzyme. In other words, it can perform cellular level chemical reactions. So if RNA evolved first, RNA could act both as information storage and as an enzyme protein, or something, not as a protein, but something similar to an enzyme. So what this means is that if RNA was the original coding molecule, then early on, RNA could store information and also replicate itself. And then as life evolved, as cells got more complicated, there was a transition to the evolution of proteins and then the evolution of DNA as the storage molecule. The reason that DNA is probably the storage molecule now is that DNA is far more stable than RNA. So we talked about the DNA double helix, the backbone, that sugar phosphate covalent bonds in the backbone, and then the complementary base pairing make DNA really stable. When you think about hydrogen bonds holding those double helix strands together, a single hydrogen bond is pretty easy to break, but a whole bunch of hydrogen bonds is not so easy to break. They're really strong all together. And so DNA as a double helix is much more stable than RNA. Because RNA is single-stranded, RNA is generally less stable. So RNA can fold back on itself, produce double-stranded regions, but the whole molecule all by itself is single-stranded and a little less stable. Uracil is also less stable than thymine. And so part of DNA becoming more stable was the switch from uracil to thymine. So sort of historically, meaning like a couple billion years ago, RNA was probably important as the very first both information and catalyst molecule. All right, so let's go ahead and talk about structure of RNA. Most of what you know about DNA is going to apply to RNA. RNA is made up of nucleotides that have the same general composition, so a sugar, a phosphate, and a nitrogenous base, with a couple of specific differences. So at the 2' carbon, we've got a hydroxyl group. That makes RNA a ribose sugar. Remember that DNA was a deoxyribose. So as far as the sugar goes, the only difference is the presence of that hydroxyl group. We still have the phosphate. There's no difference there. And then the nitrogenous bases are the same, except that instead of thymine, we have uracil. We still get a phosphodiester bond to form that sugar phosphate backbone. We still have 5' ends, 5' being the phosphate end, and then the 3' end being the hydroxyl group. And then we still have the nitrogenous bases sticking off to the side. However, RNA is always just a single-stranded molecule. The only caveat to that is there are some viruses that are double-stranded RNA. But as far as plants, animals, fungi, all of our living cells, they're all going to be single-stranded RNAs. DNA basically has one structure, it's that double helix. RNA takes on a whole variety of different structures depending on how it folds back. So down here, we've got an RNA molecule. I don't actually know what RNA molecule this is, but what it's showing you is that RNA can fold back based on complementary sequences. And so in biology, structure always implies function and vice versa. So the structure of your RNA molecule is going to give it specific properties for its function in the cell. For example, messenger RNA, if you remember messenger RNA from Biology 101, messenger RNA is always single-stranded and linear, which means it doesn't fold back on itself. And the reason that it's single-stranded and linear is that the ribosome has to be able to access it for the process of transcription. So we'll get to that. Translation. We'll get to that in a little bit. You don't have to know about mRNA right now, but just keep in mind that the 3D structure of RNA changes with the type of molecule that it is, and it changes based on how it folds back on itself. Flight simulator.
It's fun. We've come in and I tried one out
Did you?
last year. That's pretty cool.
All right. So there are a whole bunch of classes of RNA. The three that we are going to focus on are ribosomal, messenger, and transfer RNA. So we'll talk about the functions of all of those. We're not going to talk about them right this minute because we're going to get to them. All of these other RNAs, I gave you a handout of the other classes of RNAs, the table 10.2. So this one kind of describes the really basic function of all of these other RNA molecules. They're all involved in different things. One of the important jobs of a few of these, the microRNAs and the small interfering RNAs, these guys are both really important in gene expression. So they help to regulate if and when a gene goes through transcription or translation. So some of them regulate at the production of transcription. Some of them regulate at the sort of the step between transcription and translation. So the rest of these have different jobs in different settings, but microRNAs and small interfering are important for regulating whether or not a gene is actually expressed. I'm not going to go through all of these because it would just be a laundry list. So that's why I give you a handout. As far as what you would need to know for an exam, you need to know the functions of these. And we won't talk so much about structure of ribosomal RNA, but we will talk about structure of messenger and transfer RNA. So you will need to know the structure and function of these guys. And then when we get to, I think it's chapter 12, one of our chapters coming up, we'll talk about gene regulation and how RNA works there. So you don't need to know too much about that right at the moment. All right, so we are going to get right into the process of transcription. So from this point on, if you catch me saying transcription and translation, and you catch me and call me out, you get extra credit. So the process of expressing a, hold on, I feel like I missed, oh no. The process of expressing our genetic information is a two-step process. The first step is transcription. This is where we are initially going to get our information out of our DNA. So this is sort of the first step in getting information out of our DNA. So we're going to need our DNA template. In other words, we need our gene. So we are going to talk about what a gene looks like. There are some specific characteristics. We're also going to need our raw material. So just like DNA replication, we need nucleotides that we're going to use to build our new RNA molecule. And then we need what our textbook calls the transcription apparatus. This is basically all of the proteins and enzymes that are important for RNA synthesis. So when we talked about DNA synthesis, we talked about DNA polymerase and gyrase and ligase, all of those and big enzymes that were important in the process. So there's a lot about transcription that sort of mirrors DNA replication. And if we think of RNA as our early molecule, then that means that transcription and translation would have also sort of evolved out of the same type of process. All right, so we are going to cover the process of gene expression. When the fields of genetics and molecular biology, mostly molecular biology, were really, really young, we had what we call the one gene, one protein hypothesis. So this was the idea that there's a single gene that codes for a single protein, and one gene gives you exactly one protein. And then as we learned a little bit more about what's actually going on in the cell, we learned that one, sometimes genes can make multiple different types of proteins, and then two, that not all genes encode proteins. Some of our genes encode an RNA product that has a specific function. So Now we consider the process of gene expression in one of two ways. One is gene to polypeptide or functional protein, and the other is gene to an RNA with a specific function, because all of those RNA molecules that I showed you on that previous slide, they don't go through that last step of building a protein. Once those RNAs are built, then they are what functions. They don't necessarily code for a protein. So the process of transcription is what's going to get us from our DNA to our RNA. No matter what type of RNA it is, it's going to get us from DNA to RNA. Then, if we're going to make a functional protein, say an enzyme in our cells, we're going to need to do another step. And that is where the messenger RNA is used in the process of translation. I didn't even give you an option to catch me on that one. From messenger RNA through the process of translation, this is where we get a protein. So we're going to go through both transcription and translation. We're going to assume that we're going to build a protein, but just keep in mind that for all of those other RNA molecules that we talked about, they are only undergoing transcription. We are going to talk about transfer RNA. Transfer RNA would be an example of a direct-to-function RNA product. So we would use a gene that encodes the sequence for the transfer RNA, and then we don't make a protein out of it. The transfer RNA itself has a job in the cell. This process of gene expression, sometimes you'll also see this called the central dogma. So Watson and Crick both continued working in molecular biology and biochemistry. And shortly after the publication of their sequence, they started doing work on basically like, what can we use that DNA for? And so one of the next papers that came out within a couple of years was sort of an outline of a process that would lead from DNA all the way up to the protein. And they proposed this idea of an intermediate molecule, which turned out to be messenger RNA. So sometimes, especially in older textbooks, you'll see this central dogma. That's what this process of gene expression used to be called. And sometimes it's still called that. So if you watch like a video or anything or a different source and you see central dogma, that's what that is. I don't know if our textbook, I can't recall if our textbook uses that term or not. Okay. So we are going to start with a DNA template. And we are going to use that template to make an RNA molecule. So no matter what type of RNA we're making, we're going to use a specific sequence within our DNA. We are mostly going to focus on the production of messenger RNA. So that's what we're going to be talking about. But again, all those other types of RNA are produced the same way. So we're going to start with our double-stranded DNA. We've got our double-stranded DNA. And remember that our DNA runs anti-parallel. So once we define the location of a gene, we need to figure out which of our strands we're going to use as our template for making our messenger RNA. So both of our DNA strands can be used to encode genes, but remember that our sequences are complementary. They are not the same. So if you sequence one strand of DNA, you're going to get a different sequence than if you do the other strand. So for the process of transcription, you are always only going to ever use one strand. Now, which strand that is depends on the gene, but no matter which gene we're using, we are going to use what we call the template strand. So the template strand is going to be the strand that runs 3' to 5'. Now, this is because our RNA polymerase, just like DNA polymerase, builds its molecule in a 5' to 3' direction. So in order to get our messenger RNA in a 5' to 3' direction, we have to use the 3' to 5' strand. So when you are working on your problems, like in your packet that I handed out for you, when you're working on your problems, you are going to usually be told which one is the template. I usually don't ask you to figure it out, but you want to make sure that you always use that template strand, because if you use the other strand, you're going to get the wrong messenger RNA and a completely different amino acid sequence. So because we're making only single-stranded mRNA, we use only one strand of our DNA, and that's our template strand. The other strand, just to make things confusing, is called the coding strand. And to me, these words mean exactly the same thing. So you just have to remember which one is which. Template is the one that you're going to build from. When we start looking at sequences, you'll notice that the reason the other one is called the coding strand is that your coding strand is actually the same sequence as the mRNA due to complementary base pairing, except for uracil. So the reason that the other strand is called the coding strand is that it is the same code as the messenger RNA, and the messenger RNA is the one we use to make our protein. Again, I think it's really confusing. I don't know what other words they would use instead, but just keep in mind, you're going to use a template strand. So if you have your packet that I handed out last week with you today, if you look at it, can I hold that? If you look at it on all of the problems for transcription, it'll actually say template or coding. So you just want to make sure that you use that template. All right, so we need our template DNA, and then we need all of the accessory things for our, or I'm sorry, I jumped ahead of myself. Within the template strand is what we call the transcription unit. So the transcription unit is a DNA sequence. The transcription unit is a DNA sequence that contains your gene. So your gene is going to be found within that transcription unit on the template strand. Now, not all of the sequence that is part of our gene is going to end up in the messenger RNA. We have some extra things that surround our gene, but are considered all part of what we need for building our messenger RNA. So that is the promoter region, the RNA coding sequence, and then the terminator sequence. I feel like my slides are a little out of order, but that's okay. Okay. So the general idea here is that we are going to use one of our strands. So just like DNA replication, we are going to open up our double helix. And then we are going to start copying only one of our strands. So remember, DNA replication, we opened up the helix and then we were copying both strands. So for transcription, we're just going to copy the one strand using that template. We're going to see in a minute that RNA polymerase is kind of a one-man band. It does everything that all of the factors in DNA replication did. So we had our DNA helicase that opens up our DNA, and then we had DNA polymerase, and we had all these different things. RNA polymerase does it all. Which is kind of another reason that we would think that RNA was our original molecules, that the RNA polymerase does all the things. And now we have specialized enzymes for DNA replication. Okay. So in bacteria, we have basically one side of our DNA is, hold on, I want to say right now. What I meant to say is that in some organisms, genes are transcribed on one strand or the other. And then in others, they can be transcribed from the opposite strand. So if we look at this sequence here, we've got sequence for A is being transcribed from the bottom strand, C is being transcribed from the top strand, whereas B is being transcribed from the opposite one. So there's not always a single direction where all of the genes are going to be coded from. Either strand can code for your genes. Sometimes they also, just to be more complicated, sometimes they overlap. Usually in bacteria, they don't overlap, but sometimes in eukaryotic cells, they'll overlap. Okay. So let's go ahead and get into what's actually happening during transcription. So we mentioned that we need the DNA template strand. And then we need, within that DNA template, we need the transcription unit. So the transcription unit is basically all the information that you need, all the sequences that you need to actually do your transcription. So there are three main parts of the transcription unit. The first part is the promoter region. So the promoter region is a region that is going to be the same no matter what gene you are looking at. So in eukaryotes, and then even more specifically in each type of organism, like say mammals or even say humans, sometimes you will have a promoter that is specific for those particular groups. So this is an evolutionary adaptation. And what this does is allows your RNA polymerase, the same RNA polymerase, to always recognize the start of a gene. So your promoter in eukaryotes, all eukaryotes are going to have a very similar promoter. They're going to have a very similar RNA polymerase. From an evolutionary standpoint, this is selected for because if you have a mutation in your promoter region, then that gene's not going to get turned on, and then it's very likely that the organism is not going to survive. So this promoter region for any particular species is going to be the exact same, so that RNA polymerase can recognize any gene. Yes.
Okay, this might be like a duh kind of question, but I'm just trying to like organize all this information. So the leading and lagging strand that we talked about last time, that is to make DNA. And then the mRNA with the template strand, that is to make RNA.
Correct.
Okay, cool.
Yes.
Anything
And there, because RNA is always single-stranded, there's none of this like leading and lagging
Yeah.
business. It's
Okay.
just one direction, one strand.
Okay.
And it
Got it.
doesn't matter, the point of the previous slide was that it can be either strand.
Okay.
Whereas in DNA replication, depending on where in that replication bubble, force the replication to be a little bit different. But here, it's always just one strand, one direction. Your template is your 3' to 5' side of the DNA. Your messenger RNA comes out 5' to 3'. Yeah, not a duh question. Not a duh. Okay. So after the promoter, or actually, let me back up one more thing. The promoter is part of its job is regulation. So no gene is going to be copied into messenger RNA until the gene is, I'm going to say air quotes, turned on. And it is turned on when RNA polymerase binds to that promoter region. And so in chapter 12, we are going to learn a little bit about gene regulation, where the cell can actually control which genes are turned on and off at what point, because not all genes are expressed all the time, and not all genes are expressed in all cells. So for example, there are going to be genes in your neurons that are different than the epithelial cells of your stomach, simply because they have different structures and different functions. Likewise, you're going to have different genes turned on depending on your age. So if you are a developing embryo, there's going to be different genes that are turned on than when you are going through puberty, and different genes than when you are done with puberty. So depending on where the organism is, that can control what genes are turned on and off. So after the promoter, we have the RNA coding region. This is probably what you're thinking of when you think of a gene. This is the part that is going to be actually copied into the messenger RNA. So the promoter region is not. The promoter region is where the RNA polymerase is going to bind. And then just after the promoter region is going to be the start of your transcription. Transcription is going to happen in your RNA coding region until you get to the terminator sequence, which is basically the stop. So there's going to be a sequence that signals for RNA polymerase to stop copying the mRNA and disassociate. So it's sort of like having a capital at the beginning of a sentence and a period at the end. The promoter and the terminator designate where your mRNA starts and where it stops. And then that promoter also designates when it starts or when it stops.
So the strand right here, the RNA, is it typically mRNA?
This one?
Yeah.
Yes, this is messenger. Well, so I'm going to say yes for our purposes, because we're going to take our messenger RNA and we're going to build our messenger, go through transcription and translation. So yes, not always. If it is one of those other RNA molecules, then it's just whatever RNA molecule it is. Yeah, so like if it was a microRNA or small interfering, even a transfer RNA, it would be one of those. So we're going to act like they're always messenger RNAs, but they're not. Any other questions? Yes.
Okay, this is where now I'm gonna get so confused. So upstream, is that,
Go ahead, ask your question. I didn't
I-
explain it, so.
no,
go ahead.
I always get confused on which direction is what.
Okay, so Lindsay's asking about this upstream and downstream. Okay,
Mhm.
so from your coding region, upstream means before, downstream means after.
Okay.
So towards the promoter.
Yes, so yes, you would say your promoter is located upstream of your coding region, and your terminator is downstream of your coding region. I won't use that term very much just because I don't teach it in 101. So it's not like, I know it's there, but I don't say it because I don't teach that term. But yes, upstream means before the coding region. And then you'll notice here, we've got the template strand, that's the 3' to 5'. And then the non-template or your coding strand is the 5' to 3'. So you will want to pay attention to how it is written. So for example, in your packet, just, I don't know, because I didn't do it on purpose, in your packet, the template strand is on the top. In our diagram here, the template strand is on the bottom. So it's not top or bottom though, it is which one is the 3' to 5' is your template. Thank you for asking about upstream and downstream because I forgot.
You're welcome.
I forgot that it was there.
I just know that our book is gonna use it, and then it's gonna use it in like our Achieve assignment. I'm gonna get confused.
Yes, yeah. So you can also refer to like the location of genes. So in this image, you would say gene A is upstream of gene C, or gene B is downstream of gene A. So it's not just within the gene, it is all references along a chromosome. Okay. So while we were talking about transcription and translation, we are going to go through 3 steps. Both of them have initiation, elongation, and termination, and they mean exactly the same thing in all of them. We did not use initiation, elongation, and termination in DNA synthesis, but the same idea is kind of there. Okay, so to get started with transcription, of course, we need our template. And then we need our nucleotides. So just like DNA, we need our raw nucleotides that we're going to add in. Just like DNA polymerase, RNA polymerase is going to add those nucleotides to the 3' end. So they both work the same way. RNA polymerase is a really big molecule, just like DNA polymerase is a really big molecule. In bacteria, the RNA polymerase has 5 subunits. So the DNA polymerase, remember, was also a real big enzyme. We call these really big enzymes holoenzymes or holoenzymes. And then we need something called a sigma factor. So in a little bit, we're also going to talk about transcription factors. So I'm just going to put transcription factor in parentheses because they kind of do the same thing. So the sigma factor for transcription and transcription factors for, oh, nope, sorry. I'm going to screw myself up all day today. Okay. Sigma factor is also known as a transcription factor. It is something that RNA polymerase is requires for the process. The reason I'm getting tripped up is because there's more things in this than I'm used to lecturing in 101. And I just lectured in 101, and so now I'm getting confused. Okay. I will attempt to get on the right class page. Okay, so this sigma factor is basically going to act as a guide to bring RNA polymerase in. So it's going to sort of signal for RNA polymerase to come bind to that promoter region. I will endeavor to not confuse you all. It's not working. It's already not working.
There's still time left.
Shooting for the moon today. Okay. All right. So our sigma factor is going to bind at the promoter. Again, the promoter sequence is something that is similar across a variety of organisms. So when we see sequences that are the same across a variety of organisms, we call them consensus sequence. meaning they are conserved or the same across different organisms. So these sequences, these conserved sequences, are sometimes used to determine the like how organisms are related. So for example, our ribosomal RNA sequences, those are conserved sequences. So for example, all eukaryotes have the same ribosomal RNA sequence. Usually these consensus sequences are things that are evolutionarily very important. So if you get a mutation in your promoter sequence, you're not going to get RNA polymerase binding, which means you're not getting gene expression. And so these ones don't change very often. So we are going to find the promoter sequence just a short distance past or upstream of the start site. So just before the start site. Remember, the start site is where RNA polymerase is going to actually start transcribing, but it's going to bind at this consensus sequence. In bacteria, this is called the Pribnow sequence or the Pribnow box. And it's just called the box because if we look back at our diagram, it's just a region on our gene sequence. So we call this, the technical name is the Pribnow box. You may also, I don't know if our textbook is going to say it or not, but it's sometimes also called a TATA box, T-A-T-A. And the reason that it's called that is that there are lots of A's and T's in that sequence. It's actually all A's and T's. And the reason for this is that the A's and T's are connected by just 2 hydrogen bonds. And so they are easier to pull apart than a GC-rich sequence. So at our origin of replication, we get lots of A's and T's, and at our consensus sequence in the promoter region, we get lots of A's and T's because this is where our polymerase is going to bind and where we're going to initially open up our DNA strand. There is a typo on this slide. And I'm actually not sure what the typo is. This part right here. I'm not sure what the 35 is supposed to mean. So I will have to look that up because I'm not sure what that is there for.
It was in our achieve assignment,
The 35 was?
Yeah,
but I tell you what it means, absolutely not.
yeah, me either.
But it was in there, it was
The 35?
Yes, the 10 and the 35. Because our achieve assignments are like a week before what we're actually learning right now.
No,
So-
like seriously.
So like everything that was due yesterday was all of this stuff.
Today.
Yeah.
That was not on purpose. I'm sorry.
No,
I
it's
know,
okay.
I thought it was on. That's why.
No, it wasn't. It wasn't
No,
on purpose. We got behind a
At
little
first
bit.
it wasn't. At first
Yeah.
it was like on track and I was just like, oh, it's okay.
Yeah, no, we got behind.
I'm sitting here thinking like, oh, this is a really good one, like, because of-
I know what it means. Sorry. I do know. -10. I was looking at this as like a bullet point. It's not a bullet
Oh.
point. It's -10 consensus sequence. This means
Yeah,
that it's 10 base pairs. That's where that 10 base pairs upstream from the start site. -30. I was looking.
I honestly thought this was a
Yeah.
test. Like I thought you were asking us.
No,
I'm
no,
sorry,
I
I
wasn't.
don't know.
I literally got, I'm telling you, I am just not here today. The -35, I was thinking this was supposed to say 3' to 5', and I was like, but that's not, okay, -35 means it is 35 base pairs upstream
Okay.
from
So
the start
it's
site.
plus, does that mean it's?
It's downstream.
Yes.
Oh, I get it.
So are those like the most common or can that only be used two?
So for all bacteria, they have this sequence at -35 and then that sequence at -10.
Okay, so
Okay,
only those
yes.
two. Okay, I was wondering why we only saw -10.
Yes, that's where they're located for all bacteria. But I need to fix the achieve assignments because they're not supposed to be before. I wish one of you would have emailed me about that.
It's been,
It's on purpose
it's
too.
been like that for like 2 months, I think.
Yeah.
Like, it's been going on for so long that it just felt like I was like, oh, it's just how it is.
So when the publishers sell us on this stuff, like what their intention is actually for it to be before, to get you like in the material before you come to lecture.
Which it does kind of do, like it helps, I guess.
And so I don't
Helps
necessarily
spread.
agree with that because I feel like they're asking you a lot of detailed questions before you even learn about
That,
it.
Yeah.
because when it came to these, I was just guessing.
Yeah, no, it would get to the point where I was just guessing every single option, because I was like, I don't know, it all looks the same, but also it goes into a lot of detail that we don't end up going into either.
Yeah,
It's
which
like-
would make more sense after I've lectured.
Yeah.
Yeah. Okay, let me fix that. Okay. So, we are still getting ready for transcription. So, the consensus sequence is where that sigma factor is going to bind. And then the sigma factor is going to guide the RNA polymerase in. So I like to think about this as like, if you're trying to parallel park and the other person in your car gets out to guide you in to the parking spot. That's kind of how I think of the sigma factor. They're there to like guide the RNA polymerase into the right location. So when the RNA polymerase binds, and here we've got the -35 and the -10. Sorry. Those are locations so that RNA polymerase gets to the right spot. So that the very first thing it transcribes is the very first base pair in the messenger RNA sequence. And we will see in a few minutes that the very first set of sequences is always going to be the same. It's always going to be the very first thing transcribed is always going to be a messenger RNA that starts with AUG. And again, this is so that the cell gets the start of it correct. And we'll talk more about that start sequence on the messenger RNA in a little bit. But it's kind of like how a capital letter always signals the start of a sentence. If you don't have the right capital letter, let's say we're starting our sentence with the, and we don't have a T, it's going to throw off the meaning of the sentence. So the beginning of our messenger RNA depends on where the RNA polymerase actually binds, and it binds at those consensus sequences. RNA polymerase does not require a primer. So with DNA, we had to have a little primer, whether that was PCR or actually in our cell, we had to have a little primer because DNA polymerase has to add to a 3' end. RNA polymerase does not, which is why primase in DNA replication is actually an RNA polymerase. And another reason why we think that RNA came first is that RNA polymerase can work without having a template. So it can just start adding in the complementary base pairs. Has it really been more than just the last couple weeks on the Achieve?
Yeah.
Sorry.
It's okay.
I mean, we've made it this far.
Yeah.
Why change it now?
I kind of like
I mean,
We're not complaining.
I was gonna say,
it is confusing.
it's really like,
Even if you guess, you can easily get an A.
It is confusing, but like you watch the videos and I guess it kind of gets you familiar on the topic and
Yeah.
you come in the
Yeah.
lecture and you
Yeah.
get some of the stuff.
Because I just take notes of it, even if I, guess. I'm like, okay, we'll just put this down.
yeah, like it's not a big deal if
It is what it is.
Like it can stay the same as it is now and it'd be fine.
Yeah.
Okay. Maybe I won't mess with it. I don't know.
No,
I'll
you
look
don't
at
need
it.
to.
Yeah.
Okay. All right, so initiation, our sigma factor. So this slide says elongation, but it's kind of both initiation and elongation. So what's going to happen is we've got our promoter sequence. The sigma factor is going to come in and bind to that promoter. And then it's going to guide the RNA polymerase in. So first two steps there are actually initiation. And then the RNA polymerase is going to open up the double helix and then just start adding in nucleotides. So once everything is bound, we've got our sigma factor, our RNA polymerase, and our nucleotides, then RNA polymerase can start doing its job, which is to add in the complementary base pairs. So we are going to add in our complementary nucleotides based on the same base pairing rules as DNA. So anytime we have a G, C, we're going to add in the complementary pair. If we have a T, we are going to insert an A. And if we have an A, we are going to insert a U. So as you are working on your assignment, if you don't get in the U's, it's going to be pretty self-correcting because then you're going to go try to do translation and it's not going to work. But I've noticed that a lot of students get hung up on that U. And when I ask in a test to make DNA, a lot of students will put in the U because they know that U is really important for transcription and it just kind of gets put in there. So just keep in mind that U only shows up when we're making our RNA. So as RNA polymerase is working, it's going to start adding those nucleotides in. And what's going to happen is that because it's single-stranded, we're basically going to get almost like a ribbon coming off of the transcription fork or the transcription bubble. So as RNA polymerase moves, it's keeping that bubble open, but as it passes, it's actually going to let the double-stranded molecule come back together, and the RNA is going to kind of fall off out of the outside. So that's what this image here on this side is showing. It's showing that we're building this RNA polymerase, and as the double helix closes up, that messenger RNA gets pushed off.
So it's step C and D elongation and E is termination.
No termination yet. So E is just showing that we're continuing to build that molecule. Yep, so these ones are elongation. Bless you. Thank you. All right, so it's time for a break, but I would like to go ahead and get through termination before we do that. So with DNA replication, there's not really a termination. You basically replicate until you get to the other side of your bacterial chromosome, or until you get to the next replication bubble that is up or down from the one you're in. So there's not really a termination necessarily. But with both transcription and translation, we have a specific process for termination. Our textbook only goes through bacterial termination, doesn't get into eukaryotic termination, but there are two ways that bacterial cells or bacterial genes undergo termination. They are called Rho factor dependent or Rho factor independent termination. All right, so in rho factor dependent termination, there is a small molecule called the rho factor. And basically what happens is once the RNA polymerase encounters the termination sequence and transcribes it, this little rho factor, this little bubble here, this little rho factor will bind to the termination site. And basically what it does is sort of jams up the system. So RNA polymerase can't continue working because that rho factor has actually bound and basically blocks RNA polymerase from continuing. So what it's actually doing is this rho factor is actually acting as sort of similar to a DNA helicase. And it is basically unwrapping the DNA from the RNA at that site. So it recognizes the termination point and then separates the DNA from the RNA. All right, so our other type of termination is rho independent. So this one is basically a structural issue that comes up in the messenger RNA. So if rho independent termination is being used, there is a sequence as part of the terminator sequence in the transcription unit that will actually cause the RNA molecule to fold back on itself. So we talked about RNA being single-stranded, but sometimes it folds. If rho independent termination is being used, then the RNA molecule, once it gets long enough, once it gets that termination sequence, actually bends back on itself and sort of forces RNA polymerase to let go. So rather than an outside complex coming in, the molecule that's being produced itself folds in on itself and basically disrupts the ability of RNA polymerase to keep going. So if we look up here at our DNA template, we've got these complementary sequences up here. So once in our termination sequence, once we get both of those, the molecule folds back on itself and then RNA polymerase can't keep going. All right, so this is the end of transcription kind of proper. Let's take a break, and then after the break, we're going to talk about what happens to eukaryotic genes next. If this was a bacterial gene, translation can actually start immediately. As soon as the messenger RNA comes off, the translation unit, the ribosome, and the other pieces of the ribosome, or I'm sorry, of translation, can actually start immediately transcribing that gene. And that's because there's no no nucleus. The transcription factors and the translation factors are all present. And so transcription and translation can actually happen at the same time in a bacterial cell. For eukaryotes, it's a little bit different. And we have to take that messenger RNA from the nucleus out to the cytoplasm. So there's some extra steps before we can get to translation. So let's go ahead and take a break. And then when we come back, we'll talk about those differences with eukaryotic cells.